Good morning, everyone. Happy Friday after Halloween. Um, I hope everyone had a lot of trick-or-treaters, maybe not eat too much candy or a lot. It's Friday. We're heading into that part of the year where days go really fast. So we probably need all the sugar and energy we can get. So we appreciate your spending your Friday morning with us. As a reminder, this is the Team Research Forum. We host this as a virtual event about every other Friday, always at 10:30 and always online. If you are part of the audience, we're going to ask you to please make sure you keep your microphones muted to reduce background noise. And it helps to keep your video off just to get good quality image on your end. If you have any questions as we hear from our speakers, I invite you to put those questions in the chat box and we will have time for Q&A at the end. So we're going to go through all the presentations today and then we're going to try to save like maybe 10, 12 minutes at the end for Q&A and conversation. So today I am very honored to host our colleagues from the Mouse Biology program. The Mouse Biology program is quite a unique resource that we have on campus to help all of our researchers working in a variety of areas related to human disease and health and biological processes. So today we're going to hear from 4 of the mouse biology researchers or people who have made use of the mouse biology resource on campus and how it has helped advance their research program and maybe encourage you and inspire you as to how you can connect both with them and with the mouse biology program as a resource. So to get us started today, it is my honor to introduce Distinguished Professor of the Department of Surgery, Dr. Kent Lloyd.
Well, thank you very much, Ana Lucia, and welcome everybody. Hopefully you can see my screen. I just wanted to take a few minutes to introduce all of you to the Mouse Biology program. For those of you who may not know, we've been around for 25 years, if you can imagine, and we've been really trying to do our best to serve the campus community since then. In terms of the who, what, where, when, why, and how about mouse biology, well, this is it. In essence, we provide hopefully a one-stop access to all the resources, services, and training that you would need in terms of using genetically altered, surgically manipulated, and environmentally challenged mouse models for research. So we have a number of scientists, technical and staff and students. We're out of the Office of Research. We're in a number of facilities, and we support research and training. And we provide these services on a fee-for-service basis. Here's our locations, primarily off campus between the UC Davis and the health system campus is where our operations are, and we have one vivarium there, and we have a number of vivarium that we oversee and manage over on the Davis campus. We do a lot of mouse production, and over here, I'm just trying to show you a selection of the kinds of things that we can do. This is a very dynamic program. We are expert in many of these areas, and as new technologies come available, we also try to adapt those and implement those in our program as well. So that was for production, and then once you make a mouse, you need to study it, so we provide a whole host of phenotyping tests and procedures that you can use in order to evaluate over, the 12 body systems, both on a basal and on a challenged conditions. So over the last year, we've served over 450 scientists, 60 of which are UC Davis, and responded to over 600 orders for almost 9,000 products for almost 42,000 services. That's why we're so tired. We participated in over almost 200 publications over the past year in helping institutions, investigators at 258 institutions in 22 countries around the world. I want to point out a couple of new phenotyping features that we offer now. We have a home cage monitoring and phenotyping cage, which we're able to keep the mice in their home cage environment 24/7 for doing a lot of behavioral analysis and workup. And also, we just obtained a lung functional analysis system where we can do whole body plethysmography in the understrain conscious mouse, as well as respiratory mechanics analysis in anesthetized intubated mice. Well, how can you use this, and what do you use this for? Well, you can use it for consultation, requests of a service. We can execute the project for you, and we can also follow up. And the good thing about the program, I think, is you can enter anywhere along this continuum, and importantly, you can exit anytime you want. So it's all designed to be customized, flexible, bespoke to your needs. And on top of that, there's many value-added services we provide you, everything from writing up the IACUC protocol to helping you with writing up your papers or your grants. There's no requirement for or expectations that we would be co-authors on any grants. All we ask you to do are publications. All we ask you is to acknowledge us in any publications. So how do users benefit from Mouse Biology program? Well, I believe we add value to your research through providing unique knowledge, scientific expertise that you may not have in your own laboratory, comprehensive array of services, timeliness, responsiveness, unique specialized infrastructure, lower cost to academic users. We have a national, international reputation, and of course, provide a lot of letters of support. Now, hopefully this isn't too crude, but everybody's interested in costs. So here's an example. Dr. Smith needs a conditional knockout mouse for her NIH-funded research project. Well, if she tried to do that in her lab herself, we estimate it would cost her about $93,000 when you consider hiring a person, all the instrumentation and all that she would need to do in her laboratory herself. If the mouse is available from a retail outlet, you might end up paying $34,000 for that. But at Mouse Biology, you probably only pay about $12,000 for that. So using Mouse Biology with this particular project, it'd be a 90% saving over DIY and 65% over retail, like places at Jackson Laboratory. Mouse Biology program also extends access to all sorts of mouse resources everywhere, including, you're going to hear about one of them, the MMPC Live program. That's what Sean Adams is going to talk to you about in a few moments. But we can really provide you access to resources in mice all over the world. So bottom line is, what is Mouse Biology program to me? Well, it can be a service provider to you. And you can just come to us and say, make me a mouse. Or it can be an extension of your laboratory. And that is, help me to study this mouse. Or it can be your virtual laboratory. And that is, conduct a study for me. We can do any of these things that you like. So with that, I want to thank you very much for listening. I'm going to turn this over now to our next speaker. And here are all our speakers for today. And these are all investigators. I wish I could say they were mouse biology investigators, but they're not. But they've used the Mouse Biology program over the past year or two. So I'll turn it over to now Dr. Athena Salika for her presentation. Thank you so much.
Okay. Can you see my presentation? No.
Yes.
Are you seeing the speaker note or the actual presentation? I had a
Regular, like the normal.
The normal? Okay, great. So thank
So
you.
you should press the from beginning, I think, top on the left.
Okay, I am.
And then switch it. And then go to.
So it's not, it's swap.
Yeah.
Now it's correct?
Yeah.
Okay.
That's correct.
Okay, great. Well, thank you for inviting me to this presentation today. My name is Athena Salika. I'm from the Department of Dermatology. And I want to start by saying that I'm a big fan of the MVP of this program. Not only, I mean, it has helped me tremendously, and I know a lot of scientists have benefited. And as Dr. Lloyd mentioned, it put UC Davis on the map, both nationally and internationally. So today I will talk to you only about a small project that I work, that they help me with. In my lab, we work in burn wound healing and in neuroinflammation projects. We mainly study how immune responses affect tissue repair, and we're focusing mainly on myeloid cells, and we rely on MVP in almost all of our projects. Today, I will talk to you about our burn wound healing project. It's the most recent project that we have worked with MVP. Just to give you a little introduction on burn wounds, you probably know they're very difficult to heal. They're characterized by high mortality rate, require excision, grafting, and rehabilitation. And they can be highly inflammatory wounds, and that's how they differ by other wounds. And this can lead to increased scar formation and debilitating scar formation. So understanding the signatures of burn wounds, it's important to understand the differences from other wounds and how which pathways we can manipulate to achieve better healing. So we use an animal model, a murine animal model. We use both male and female mice, which they undergo scalp burn injury. And we isolate various tissues at various time points, even after wound closure, to look at wound maturity. And we analyze by histology, immunohistochemistry, flow cytometry, RNA-seq, and lately ecozoonoid analysis. And this is just to orient you a little bit on the wound healing process. The first, there are three phases. The inflammatory phase, which starts very early after the injury, and it consists of infiltration of the tissue by immune cells, mostly neutrophils and monocytes that they locally become macrophages, inflammatory macrophages. And the goal of this phase is to sterilize the wound, but it can cause secondary damage if it's not controlled. And after that, the proliferative phase, as the name suggests, it's a phase that is characterized by increased cellular activity, proliferation, and the goal is to make the cells that are necessary in order for the wound to close. And there is a remodeling phase, which is an attempt of the tissue to restore the original architecture. And this is a phase that fails in most wounds, and specifically in burns. So all these phases have been characterized in excision wounds. And in burn wounds, they are distorted. There is a highly inflammatory phase, which affects the proliferative phase and also the remodeling phase. So another introduction I want to make is on the molecule that I will be talking to you about, which is 5-lipoxygenase. 5-lipoxygenase is the protein, and the gene name is arachidonate lipoxygenase 5, or ALOX5. It is an oxygenase within a larger family of enzymes, and their main job is to metabolize polyunsaturated fatty acids. This is probably a diagram that you're familiar with. It shows how arachidonic acid can be metabolized by various enzymes, such as within the cytochrome P50 family, the cyclooxygenase family, and then the LOX family. As you see, there are about 4 members, but we are going to focus on 5-LO, which is an enzyme that is necessary for the synthesis of leukotrienes. And this is just a smaller schema of 5-LO pathway. As you see, during activation, this pathway generates two families of leukotrienes. One is LTB4, which is known as a potent chemoattractant and activator of leukocytes. And then 3 leukotrienes, the C4, D4, and E4, that they're collectively known as cysteine leukotrienes. And they have been identified for their ability to promote proconconstriction and vascular permeability. And of course, 5-LO leukotrienes were initially associated with asthma and atopy disease, but we now know that most inflammatory diseases are associated with 5-LO activity is associated with a lot of inflammatory diseases, cardiovascular disease, infection, cancer, neurodegeneration, etc. And leukotrienes are found in low levels in many tissues with unknown functions, actually not well-defined functions. And we found also that they are expressed in low levels in steady state in skin. So we examined 5-LO during burn injury. What you see here is a few images of skin. On the top, you see the neoepithelium. That means the new epidermis that is being made after a burn injury. And on the bottom, you see the wound bed. That is the part of the dermis that is infiltrated by immune cells. And as you see here in this image, we have stained with red keratinocytes, and there is only a couple of keratinocytes that we found here and there that express 5-LO. The rest of the signal in the epidermis was coming from Langerhans cells, so they expressed a high level of 5-LO during steady state and in burn wound healing. And within the wound bed, the majority of 5-LO was found on myeloid cells that they were infiltrating the wound, or local myeloid cells. And a large number of these myeloid cells were neutrophils. We also examined the products, and we saw that both LTB4 on this arm of the pathway and CCLTs, this part of the pathway, are found in the wound. Interestingly, LTB4 is found early in the wound, early after burn, while CCLTs slowly are synthesized slowly after burn. They initially will lose CCLTs, and they slowly upregulate during the later stages of wound healing. And that's when we went to MVP. We were actually making this mouse for a different study. We used the knockout first allele design, which is a wonderful design. It allows you to get from the single founder to get the global knockout and the floxed form of your allele, which you can then bred to whatever Cree you're interested in to get cell-specific or time-specific deletion. We confirmed that these mice did not make 5-LO, as you see here in immunohistochemistry. You can see very nicely 5-LO both in the dermis here and the epidermis after burn injury, but it's not there in the 5-LO knockout mouse. And when we looked at the products, as you see here in this color, there were very, very little or no product detected. So we're very confident about this mouse, and we examine it in burn wound healing. I will be talking to you mostly about reepithelialization rate. This is a way to analyze wound healing, and what you see here is a wound, a histological analysis of a wound. This is how the normal skin would look like. This is the new skin that is generated, and this is still unhealed wound. So when we did this analysis, we saw that the knockout mice had increased reepithelialization, that means increased wound closure compared to the controls. There was a slightly decreased number of overall cell infiltration at the wound site, and we didn't see a difference in reepithelialization in later time points. We analyzed these mice by RNA-seq analysis, and we did see that they had, in the early time points, they did have decreased pathways that were associated with immune responses were decreased. However, at the later time points, we saw that they had decreased collagen formation and decreased, which would associate with decreased healing. So we went back to our original data, and as you remember, I told you that we had LTB4 production much earlier compared to CCLT. Actually, CCLTs are almost absent during the early times after burn injury. So we posed the questions: do the different leukotrienes play different or opposing roles in burn wound healing? In order to do that, we use specific inhibitors. Actually, Berstatin is not super specific, but it was the one that we had easily access to at that point. We used inhibitors that block each pathway. So when we block the LTB4 pathway, we saw increased reepithelialization. This was associated with less infiltration of the wound, no difference in reepithelialization at later time points, similar to the ALOX5 knockout, actually. But when we block the other part of the pathway, we block the signaling of the pathway by blocking the receptor using a clinically used antagonist, we saw that we had a decrease in reepithelialization. That means and impair wound healing. And it was a much more pronounced effect. There was no difference in the infiltration of immune cells, but we had delayed angiogenesis and delayed collagen deposition. So then we came to this model in which we hypothesized that the increased levels of LTB4 during the early stages of wound healing promotes the inflammatory phase, and this may delay healing, while the cysteine leukotrienes that are being synthesized during the later stages may support the proliferation, the proliferation of the cells during wound closure. We also wanted to assess the cellular source of LTB4 or CCLT. The reasoning was, if we can shorten the inflammatory phase a little bit, do we allow for CCLTs to promote healing? So when we looked at the cells that are in the wound and they may have make leukotrienes, there are numerous cells that can make leukotrienes. There's question mark on fibroblasts and keratinocytes that I'm not going to go into right now, but we zeroed in on neutrophils because neutrophils can only make LTB4. They cannot make other leukotrienes. However, they can help other cells make leukotrienes. And there were many studies that showed that neutrophilic LTB4, that means LTB4 that's made from neutrophils, is associated with neutrophilic chemotaxis and swarming of neutrophils in other inflammatory models. So we were pretty confident that neutrophilic ILOX5 or 5-LO will mediate the inflammatory effects of 5-LO in burn wound healing. So we went back to MVP. We crossed our ILOX5 flox mice, which we made with them, with neutrophil-specific CRE, and we saw that we had the hypothesis that neutrophil-specific knockout mice should have drastically reduced neutrophilic infiltration, earlier wound closure, and characterized by increased reepithelialization. So here is the mouse that we made. In red, you see neutrophils at the wound bed, and in green, you see 5-LO. This is a control mouse, and you can see that many red cells express green, so a lot of neutrophils express 5-LO. But in a neutrophil-specific ILOX5 knockout mouse, we saw neutrophils, but they did not express 5-LO. So we were very confident this is a very good mouse. However, when we looked at reepithelialization, not only we didn't see increase, but it looked so far that we have decreased reepithelialization in these mice, and we didn't have any difference in neutrophils. So our hypothesis was nullified. It's still very interesting, and we're still trying to understand why this is happening. We have much more analysis underway, and specifically, we want to understand the eicosanoid environment at the wound site. We may need to go to target another cell type, in which case we will go back to MVP for another CRE. And in summary, I think I showed you that 5-lipoxygenase exerts dual and likely opposing function in burn wound healing. Blockade of LTB4 pathway promoted wound healing, but it doesn't look like it's likely to be through neutrophils, which is very interesting if this pans out. Blockade of the other pathway, the CCLT pathway, impaired wound healing, and our preliminary data suggests that the CCLT signaling can be a pro-proliferative or pro-fibrotic signal. So here, I want to thank you, the people that helped me do all this work. Of course, the student in the lab. This project started with Alan Nguyen, and now it's continued by Shannon. Cayman Chemicals, DNA for technologies here at UC Davis. And all these people helped me with bioinformatics support. And of course, Dr. Iserf and Dr. Murphy that always helped me with any of my projects. And here, a big thank you to MVP. They have been amazing from the beginning to the end. Brandon Willis, I know he's not there anymore. He helped me with the construct design. General help I got initially from Samra and then from Renee, who I realized I understand she's retiring, which was a bittersweet for me. She had helped me so much since she started in every level of my project. and I wish her the best in her retirement. I'm sure the next person is going to be just as helpful. We also use other facilities at MPP, cryopreservation, derivation, and import-export. We are located at in Sacramento, so we continuously get the mice from Davis. And of course, I want to thank the help that I got with the colony management from Jessica, who's now not with MPP, but also from Sarah and Erica. And last but not least, my collaboration with MPP led me to a few grants, and it's not just by this project. There were a few other projects that I worked with them on in neuroinflammation and wound healing, and also helped me help 3 graduate students at UC Davis graduate, and a few more that are going to start, and multiple collaboration. So with that, I am done.
Thank you so much. We're going to move on to our next speaker. I want to introduce Dr. Sean Adams. He's the Vice Chair for Basic Research at the Department of Surgery, and he's going to talk to us about the MMPC. I'm going to ask our next speakers to please try to stay to time because we have about 30 minutes left and 3 presentations.
Got it, Ana Lucia. Thank you. And Athena, that was amazing. I've never seen you speak before. I want to see more. That was awesome. Yeah, I want to echo what Athena said. I've used the mouse biology program for our laboratory as well to make some knockout constructs and things of this nature. And it was just absolutely, fabulous from A to Z. So, and led to a lot of really interesting high impact data and papers. So. Today, though, my job is to talk about the MMPC Live Vibrant Partnership Program and the MMPC Live Mouse Metabolic Phenotyping Centers Consortium. So I will not take much time, but I will kind of give you an overview of these. And of course, there will be time to do Q&A, and anybody can reach out to Kent or I about this after the fact. There are a number of MMPC team members on this call, I see. And they're all to be acknowledged here because none of this could come together without all of their help. So I think I don't have control over this. Is that correct, Ana Lucia? So I'll do the next slide thingy. All right. Yeah, so what is the MMPC, the Mouse Metabolic Phenotyping Center? It's been around, Kent can correct me, but I think we've been around here on campus for, I think, 15 years plus in one form or another. And in a nutshell, the Mouse Metabolic Phenotyping Centers group or consortium is nationwide and can do a whole lot of things for people to phenotype their mouse. So I'll just say next slide, next slide, next slide. You'll see a bunch of stuff. Microbiota, metabolic rate. We're even looking at the xenometabolome. So if you're interested in bacterial metabolites, we know how to do that stuff. A lot of behavioral parameters, food intake behaviors, endocrine inflammation factors, cardiovascular readouts, and so on and so forth. And we're always open as well to working with investigators to come up with, new ideas and new phenotyping methods. But we encourage folks to come to us early and help us to give you advice if you want it on study design and that sort of things. And then we can actually do the studies. As Kent said, we can actually do the studies for the client. Next slide. So this is really a nationwide consortium. You see us on the left-hand side there as a star, but the phenotyping centers are Vanderbilt, Yale, University of Michigan, and ourselves with a coordinating unit at Augusta University. And why this is important to folks on this Zoom is you can tap into any of these consortium members and we can help you do that. So if we don't have tests that are of interest to you or that we think, upon consultation won't help your program, these other awesome sites and world leaders can actually potentially do some of those assays and mice can be shipped to them and so on and so forth. So it's really a wonderful resource. Next slide. Yeah, as I said, most of the projects are focused on living mice. However, we can be creative in terms of how the living mouse projects are coordinated and conducted. So we keep an open mind to that sort of thing, but most of the studies are done in living mice. And as I said, we're a national consortium. We operate as partners together. In addition, the bottom points there are very important. We're here to help. So folks who want to reach out and just start to think about phenotyping a mouse can have conversations with any of us in the consortium to start the process and get some advice as to what they might want to look for. Next slide. So what about the Vibrant program? This is a really unique aspect of the MMPC Live consortium. NIH sort of tacked on to the MMPC this Vibrant program, and I highlighted the red stuff, which is the most important part of their RFA, which is essentially leveraging the MMPC Live consortium for small research institutions, for example, that have not traditionally received significant funding, or institutions that have served historically underrepresented populations or underrepresented groups in the biomedical sciences research world and research grant funding. So this is a broad swath of a remit from the NIH, and we've been actively working with the other consortium members to develop a program that accommodates these goals. Next slide. So here's what it looks like, and if it doesn't apply to you, it could very well apply to somebody you know in your department. So keep that in mind, and it's an incredibly valuable program. To be Vibrant eligible, and I'll explain the benefits of being Vibrant eligible in a minute, you need to be a new investigator, and you either need to be from a minority serving institution, and by the way, UC Davis is classified as an MSI, or be part of the historically underrepresented group in the biomedical research environment. So there's all kinds of asterisks at the bottom, and I'm happy to provide these, but we've defined this based on NSF and NIH guidelines, but this should not be restrictive. I know this seems restrictive, but we want to work with you. We want to work with early stage investigators. And we're finding that this is open to many, many individuals depending on their institution or their background. So keep that in mind. Next slide. We started this program as a partnership program out here in California. Each of the other sites have different partnerships, but we started this through a consortium of CSU system campuses, which most of these are Hispanic serving institutions. And so we are actively reaching out to these campuses to partner with us and with UC to advance their research capabilities. And for those faculty interested in NIH funding, we want to be a resource for these folks. So our goal is to provide, prioritize, and expedite research and training opportunities for the institutions, any faculty at these institutions who are early stage investigators. Next slide. Ultimately, we want to enhance competitiveness for research through NIH and our funding. So what's in it for somebody who's a Vibrant eligible client? Next slide. 50% reduced cost phenotyping. So Kent mentioned earlier the already really high value that the MBP and MMPC have to offer for you in terms of cost savings. Imagine an additional 50% off of that from any MMPC Live Center in the United States. Next. Access to national experts consulting on projects and concepts. And this is really exciting. Thank you, Kent, for this. 50% reduced cost in the MBP program. So you saw all of the really cool knockout and conditional knockouts and cell-specific knockouts and these other things. Those can be a 50% reduced cost for Vibrant eligible partners. We started a career development and travel award competition, which is going to be fired up here very soon. And this will be to help promote travel and other career development activities upon application to the consortium. Next. And I think this is the final slide. This just gives a sense of sort of where we're headed and the big promise here. Each institution here have responded to our town halls and sort of Vibrant outreach efforts over the last few months. And these in red are actually institutions who have investigators who have taken advantage of the Vibrant program. And we're only hoping this grows even more in the future. So to sum it up, the Mouse Metabolic Phenotyping Center Live Consortium is a huge resource for everybody, whether you're Vibrant eligible or not. Reduced cost, access to national thought leaders in different areas of phenotyping, including metabolism and certain other phenotyping outcomes. And in addition, for you or for someone you know potentially, this Vibrant program could be really, really valuable to give them a boost early in their career for reduced cost phenotyping as well as national expert input on their projects. So with that, I shall close up and hand the baton off to Joanna.
Hi, everyone. Oops, here we go. I am Joanna Bautista. I'm a neonatologist, and it's just kind of funny following all the things that Sean has said, because that pretty much describes me. I'm a very early career investigator, a physician scientist that came out of fellowship a few years ago and have been settling my way here at UC Davis. And so I'm going to just kind of give you guys a brief overview of my interests, because again, it's fairly early, and I'll kind of wrap it up with the things that I've been really working very, very closely with the core team right now in the last few weeks, particularly few months, actually. So my general interest as a neonatologist is the neonatal gut, and kind of, ranging from things such as necrotizing enterocolitis, which is pretty devastating in the preemie population, but even in the term population, we have a host of various congenital anomalies that have associated like motility and motor dysfunctions that tend to last throughout life. And the more we know about some of these perturbations earlier in development and in utero, the more we're seeing that there's actually more of a longer term impact in like adult health. So for me, that's kind of the bridge where I worked on a little bit more of the kind of juvenile population, and I'm slowly making my way down. But my interest kind of spans like what we can do and what we can kind of understand to better target a lot of these disorders. So so my primary interest has been piezo-1, which is a mechanosensitive ion channel, and it is actually a very important mechanosensitive channel in the rest of the body. So it's been really well characterized and defined more in the innate immunity and the red blood cells and the cardiac development, embryology. But in terms of the gut, has been less defined or less really well understood. And I think part of that is what we're learning is that there's a really big developmental aspect to it, in that it's coming out more or higher expression earlier on in life, but that has a different role later. So this is where, and then, I don't know, whoever found piezo clearly loved Star Wars, because the agonists that currently exist for it are called Yoda and Jedi. And so hence you'll see a little sprinkle of Star Wars forces. So in terms of kind of bridging my previous work to now, since piezo-1 is if you knock it out generally, it results in embryonic death. In humans, we end up seeing hydrops fetalis, so we also see pretty devastating outcomes. But that's what makes it really kind of a difficult mouse or difficult gene to study, because if you lose it, you're kind of stuck, right? You don't have anything to study. And so when I was in fellowship, we used the MYH11 that was unfortunately sex specific, so it was Y chromosome driven. However, we did find a really, really interesting phenotype in that we saw significant delayed gut transit, but also prolonged, weight impairment. So during this period of its life, it has other mice where they continue to have a nice trajectory. The piezo-1 knockout mice, the smooth muscle cells, had an impaired one. We looked at contractility kind of on multiple levels, and I don't have it here, but we also saw pretty significant effects on the epithelium as well as the gut muscle part. So that was also interesting, because that's something that we're only just now starting to characterize, this whole multi-signaling, multi-layer characterization. So it's a very unique niche. So it's a very hard study sometimes to tell people, but my interest particularly, right, is going back to the development. So when I thought back in fellowship that P28 was an adult mouse, when I actually looked at it on when they've shown comparisons with the human neonate, P28 really is when the term neonate kind of has similarities in terms of their gut development. So that just means that we were hitting it at a more developmentally critical stage. Whereas in another one of my mentors had treated their mice, the same mouse model, at later in life at P56, and in adulthood, you actually had an opposite effect of piezo, where they protected against constipation or age-related motility aspects. And this is just focusing on the motility and focusing on the muscle, because we know motility is really, really important for embryonic development and later functions of the gut. That's the part for me that's actually even more interesting. So I came to, I knew about MVP because of one of my previous mentors was friends with Kent, but trying to understand and trying to navigate the two worlds. I'm, I also in the clinical world and in the ICU, but trying to be able to get some support and help was a really massive undertaking. And it really speaks, like as Sean was just explaining about the different levels of support. And even now I'm like, oh, I didn't know you guys did that too. I was like, great, because I'm struggling with my cook right now. So there are so many aspects of it that I think is so beneficial for people like me, particularly who has these really awesome prelim stuff that I'm trying to now develop and become my own thing here, but struggling. So one of the things that I was really trying to establish is the fact that I need a mouse that is the new MYH or the new smooth muscle cell driver, which is not sex specific, mainly because we know with gut motility, with anything, actually most things now, that there are sex differences. So that's part of my initial thing. And then I got it really fancy. Because then I've been meeting with Jen, Laurel, and Renee, and then I wanted to get the marker as well, the reporter, the MTMG. So all these things, seem so simple when you read it, and then it is so complicated, and they have been so patient with me and trying to break everything down for me. So I really think like that aspect you can't get anywhere else. And I've asked, I've looked around, asked previous mentors, like people don't necessarily think of the transgenic mouse model as deeply as we need to sometimes to understand it, to develop our own model. So that has by itself already been a resource for me. I understand mouse genetics way better than I would have expected. And then on top of that, the functional studies that they offer, I was so shocked when I came across it and found out how many of the things they could do, because I'm struggling to get the lab staining or running qPCR at this point. So I'm not even I can't even replicate any of the gut transit studies and the things that I had done previously in my previous lab. So the fact that it was an ability to do that without, I think Kent had mentioned previously as well, that you can kind of an extension of the lab or ability to get this prelim data, get some data while I'm waiting for other things to kind of get in place is really huge, especially here where I've been, because I've been waiting for the lab stuff to come up, I've been getting better at grant writing. And so things are starting to move, but you can only move people so much or train them so far. So I think for me, that was another actually aspect that has been a massive, massive help. And then just planning out the studies. Like I even asked them, I was like, can you guys just show me what the breeding plan is if for generational? Because I think these are tools that as I grow as an investigator, I think just we overlook these little skills that we just really don't have formal training on. And so I think that being able to use the Vibrant program and then I've just been really happy I've found it. And I do tell people all about it because I do think that it's not as well as known as it should be. But I think so far it's a massive, massive help, just the pure guidance, ability to talk to people through it and okay, does this mouse make sense? And there's so many mice models, there's so many levels of it. And I think that was so TBD, this is just kind of the beginning for me early on. And but I do really, really appreciate everything I've gotten already from the program, even though we don't have the mouse yet. We just the constant feedback and kind of helping me develop and designing experiments. So the fact and their comprehensive phenotyping is phenomenal. This probably would have taken me years to actually get all the little things done. And, the fact that they have such a strategic way to do it is pretty exciting. So with that, I just wanted to do a special shout out though, Renee, Laurel, and Jennifer for their extra time spent with me, and Sean too, I guess, because he put us all together. But but I really just want to thank everyone and kind of thank to all my mentors and all the people that have been helping me transition.
Thank you, Joanna. For our last speaker of today, I am very happy to welcome back to our forum, Dr. Amparo Villablanca. She's a professor in the Division of Cardiovascular Medicine, and she is the director of the Women's Cardiovascular Medicine Program.
Thank you so much,
I have your
Anna.
slides. Give me one sec.
Okay, great. While you do that, thank you so much, and to Dr. Kent Lloyd for inviting me to participate in this forum. I'm going to be talking about how we have been using the mouse biology program as an extension of our lab, that function of it, for ongoing studies to look at the metabolic and multiomic effect of diabetes and male-female sex on cognitive function. And I wear multiple hats in our health system, but I'm going to be speaking as a PI of the Research Center for Women's Cardiovascular and Brain Health. Next slide, please. Just to give you a little bit of background, because I know that not everyone is familiar with each discipline, obviously. Cardiovascular disease is the leading cause of death for men and for women, and dementia is currently the fourth leading killer of women, and both take a greater toll on women than men, with greater deaths in women. They both share common predisposing risk factors, including elevated cholesterol, high blood pressure, diabetes, and obesity. And therefore, both represent kind of the cumulative lifetime exposure to these risk factors, which we know are largely preventable. The progression of vascular dementia follows these predictable temporal and spatial patterns where specific brain regions are affected at different stages of the disease. And the hippocampus is a particular area of interest in our studies because it's a key memory center. However, there are a number of unknowns. For example, why the disease affects certain regions of the brain, why the microvasculature is targeted, why specific subpopulations of brain regions are selectively vulnerable. And therefore, our area of focus is working at this intersection of heart and brain health, where we're hoping to unravel the contribution of each one of these major risk factors to the dysregulation in brain memory centers, specifically studying the impact that they're having on the microvasculature and how that effect is modified by sex. Next slide, please. The primary objective, therefore, is to use murine models of the cardiometabolic risk factors that are shared between cardiovascular disease and dementia to assess their multi-omic effects and how they are differentially impacted by biologic sex. So far, we've used commercially available murine models to study hyperlipidemia, obesity, hypertension, and diabetes. And I'm going to be sharing our work using the DB-DB murine model. The goal of the ongoing study is to look at the life course exposure to type 2 diabetes on brain metabolism, on cardiometabolic profile, the brain microvasculature, gene expression in brain memory centers, focusing on the hippocampus, and how they are modulated by epoxide hydrolase inhibitors, which are neuroprotective in both males and females, so that we can assess the sex specificity and dependence. And for these studies, we're using an aging model. Our first hypothesis is that diabetes over the life course is detrimental to late life brain health, perhaps through neuroinflammation and worsening permeability. And resulting in microvascular injury and therefore impairing cognitive function. Our second is that the modulation of metabolites, as well as inhibition by epoxide hydrolase inhibitors, which are clinically relevant and in clinical use currently, can function as disease-modifying tools. And the third is that there will be sex specificity in the brain hippocampal microvascular response to diabetes. Next slide, please. Epoxide hydrolase inhibitors are a group of enzymes, there are 5 of them, that catalyze the conversion of epoxides to less active species. They have a role in conversion of bioactive EATs, and they are vasodilatory, they're anti-inflammatory, and have pro-androgenic effects, so these are good. And they offer multi-cellular protection throughout the body, including recently described neuroprotection. And soluble epoxide hydrolase inhibitors, therefore, increase epoxides, including EATs, and are in clinical use, as I mentioned. And for this reason, we're interested in testing their neuroprotective actions in multiple neurovascular unit cell types, including endothelial cells in both males and females. Next slide, please. This is a busy slide. I'll just briefly go over the comprehensive methodology for these studies, which includes metabolism, cognitive function and transcriptomics. Going along the top, we have our murine model with the DBDB aged mouse. We're trying to take these mice out to 9 months, perhaps even out to 12 months. They're fed a purified diet and given the inhibitor in the water. And of course, a vehicle control. We remove then the hippocampus from the brains, and then we either subject it to cryosection and laser capture micro dissection, and then microarray and bioinformatic analysis, or single cell, rather single nucleotide transcriptomics and RNA sequencing, and then submit that through a multi-omics pipeline where we can identify the different cell types, including the endothelial specific differentially expressed genes, coding and non-coding targets, and their targets, and then pathways and networks, and then correlate to published databases regarding differential expression in humans with dementia. On the left-hand side are the functional assessments that we're doing, and these are studies that mouse biology program is critically important for in our studies, behavioral testing, cognitive testing, and then the metabolic assessment for glucose, insulin, lipids. And we're also doing home brain metabolomics on these animals. Next slide, please. So I've mentioned already some ways that the mouse biology program is supporting our studies. We've been working with MBP for 25 years since it was first started. The staff runs our MMPC live phenotyping tests and procedures, which we just heard a little bit about through 2 of the cores, the animal core and the physiology and behavior core. They perform the cognitive testing, provide us the data and graphical summaries that I'll show you. They obtain the blood for the cardiometabolic testing and obtain body weights. They conduct the water treatment with our inhibitor, the TPPU, and also water intake measurements. They perform the colony management. They coordinate with veterinary staff their issues, and they are included in our IACUC protocols. And then importantly, they're helping us with troubleshooting because we are seeing attrition in mice during the type 2 diabetes aging study. As you can imagine, these are very sick animals. Next slide. I'm not going to go through the details of this, but this is an example of how we are, with MBP's help, showing that our model is obese, hyperlipidemic, and hyperinsulinemic. The wild type animals are shown in green and the diabetics in purple, males and females. And of course, we're confirming that model. Next slide. And then we're also assessing lipids for all of the different components of lipids, looking at diabetics compared to wild types and demonstrating again, confirming the model as being a hypolipidemic model. Next slide. And then for the cognitive testing, we're using a couple of tools, locomotor behavior by open field testing, where we look to see how much time is spent in the center of the open field, the distance traveled to the center, and the total time traveled, and showing that the diabetic mice have significantly worse performance, which basically means that they have less exploratory behavior. And next slide, using Y-MACE, which helps us assess for not only exploratory behavior, but also short-term and spatial memory. Again, the diabetic animal shown in the purple, and demonstrating that they have impairment in performance by Y-MACE testing. Next slide. This is just brief, and you can just click to fill the slide. And keep going. Thank you. Oops, just go back 1. Thank you. This is our single nucleotide transcriptomics. We don't have yet the data on the aged mice, but we do have data on our earlier studies looking at 4-week-old mice. And along the top, you'll just see that we can demonstrate with UMAP the different cell types and clusters in the hippocampus. And then with PLSDA, the different global gene expression profiles between diabetics shown in the red on the bottom left, and the wild type shown in the blue completely different gene expression profiles. And then moving to the center, the heat map revealing the differentially expressed gene patterns with mostly upregulation on the left within the diabetics compared to wild types, and then moving on to pathways of these genes on the top right, showing that diabetes modulates endothelial cellular pathways and pathway networks, and showing the upregulated genes on the bottom right in red, downregulated in green, long non-coding RNAs in yellow, targets of long coding RNAs in gray, and then transcription factors. So we can do comprehensive multi-omics. This is the male pattern, and going on to the next slide very briefly, just to show you the female pattern, which is different, both for PLSDA, heat map, and gene pathways and networks. And then going on to, I think, my last slide, which is where we're now, and we do this for all of the risk factors, look at the sex and cell-specific effects of type 2 diabetes on the entire neurovascular unit transcriptome using a sex and gender-based analysis. The males are on the left, females are on the right, and these are Venn diagrams showing endothelial cells, microglia, astrocytes, and neurons for males on the left and females on the right. And this is just showing the pathways that are unique, which are the unhighlighted for each cell type and each sex, and those that are common between males and females shown in gray in the middle part, and then the unique pathways for females for each cell type on the right-hand side. So this is very powerful, allowing us to identify really key, very important sex-specific differences in the entire neurovascular unit. Last slide is just the anticipated work impact of our work. We're hoping to provide a detailed molecular analysis of how type 2 diabetes activates an inflammatory and neurodegenerative cascade in brain microvessels and brain memory centers, focusing on the hippocampus and how that relates to other cells in the neurovascular unit, and its reversibility by soluble epoxide hydrolase inhibitors. That work is in progress, and so I don't have that to show you, but I'm hoping it will be a positive and informative. And then also, we're hoping to analyze the impact of type 2 diabetes on each cell type during aging, both for protein coding and no coding transcripts, and then to look at how sex-linked biology contributes to the molecular footprints of brain microvascular disease, and then correlate the transcriptomics to the metabolism with the help of mouse biology program data and to cognitive function. And I think that by understanding the role of how each of the risk factors contributes to vascular dementia in males and females, we may be able to identify sex-specific molecular therapeutic targets for vulnerable cell types in vascular dementia. And then the last slide is just the acknowledgements of my research team and all of the cores that we use on campus, and mass biology program, and of course Kent Lloyd, and then our funding. Whew, that was a quick review. Thank you so much. Left a couple minutes.
Thank you to all of our speakers. Very impressive work. I think we have time. We're a little bit over time, but I think if people don't mind sticking around for a few minutes, we could take a few questions. Let's see if we have anything in the chat, or if you have a question that you would like to present to the speakers, just use the raise your hand function, and then I'll call out your name. There's a question from Angela from Amparo. She's asking if these like transcriptomic and metabolomic lipidomic data sets can become available for access by other investigators, and how potentially they could be accessed.
Yes, they are. Thank you for that question, Angela. They are. We can't publish without depositing the data into the genomic databases. So they are. If you're interested in any particular one of them, just let me know and I can give you the access number for them.
Thank you.
I actually had a question for the program in general. I think this came up when I was talking to some other early investigators, but is there a way that, like, we're all, you know, if I'm generating this mouse, et cetera, right, and it hits certain other organs, and I've had people kind of interested, is there a way to like make some sort of kind of universal place? Like, if I know this person has this mouse, maybe we can collaborate and work on something together, you know, like, I don't know if there's thoughts. I don't even know if this is the right form to ask, but I'm just curious because I realized someone asked me that the other day and I forgot to.
Great question. Yeah, the first and best way to do that is if you make a mouse, if someone makes a mouse,
Mhm,
especially if you're using NIH funds,
mhm.
but even if you're not, the best thing to do is to put it into a public repository.
Okay.
That way, the public repository then takes over the outreach, raising awareness and trying to
Mhm.
get the word out about the mouse. And then when you get requests for it, you can always send a message to the whoever's requesting it to say, contact the repository. And it just so happens UC Davis has the largest academic mouse repository in the United States. Did you know that we are 6 times larger than the Jackson Laboratory?
Whoa.
Wow, that's
You
actually,
guys we have so many great things, great toys in our backyard that we don't think about it. So the MMRC is funded by the NIH. We have over 50,000 mutant mouse alleles, and we're regularly accepting mouse models made by investigators from all over the country. So we're part of a 4 repository center system in the country. So
Mhm.
that's the best way. After
Okay.
that, and if you wanted to, maybe if you weren't quite ready because you still want to maybe keep that mouse for yourself to do some of your own studies and get those nature papers and cell papers. But if you still want to get the word, if you want to hear about what other things are available, there are
Mhm.
listservs that you can look at. We can
Yeah.
provide that kind of information to you as well. But I would check out other repositories,
Okay.
check us. And we're also on social media, and there's a lot of oftentimes, LinkedIn and Twitter,
Mhm.
X, reports on mice that are available.
Cool.
Yeah.
More questions from the audience? Anyone else mutant mouse curious? I have a question. As a researcher who perhaps there might be some people in the audience thinking, oh, I might want to work with the mouse biology program and develop a murine model for my particular question. Realistically, what's the timeline from like reaching out to you and having the conversations and figuring out what needs to be done to actually having a mouse and being able to do actual experiments?
Great question. And of course the response is going to be it depends, because if let's say the mouse, just like Dr. Batista just asked, let's say the mouse already exists in a repository. Well, we could conceivably get it for you in a couple of weeks. On the other hand, if you wanted to make from scratch, a conditional inducible mouse in a particular tissue at a particular time, that could take 12 to 18 months to actually make the mouse. And then there's a lot of validation work that has to be done. So I would say anywhere between a couple weeks and 18 months, you could have the mouse that you want. It all depends on the level of complexity that you want in the mouse, but that's about the time frame you're looking at. Now, you might think, gosh, that's a long time, 12 to 18 months. But if you were to do it yourself in your own lab, it could take years and, of course, cost much more. So I would just keep all that in mind.
Well, I think that we are not getting any more questions. I learned a lot today that I did not know about the mouse biology program. I hope that everyone else who joined us today also is walking away with some actionable information. We have a recording of this session that we'll be posting online. I'm sure all of our speakers would be very happy to entertain your questions over email if you're interested in learning more about their experience with the mouse biology program or their specific research. And definitely, if you're interested in working with the mouse biology program, reach out to Kent. If we can help you with any connections related to what you heard about today, please don't hesitate to reach out to our team. And we are always grateful for ideas. What do you want to hear about in the next Team Research Forum? And with that, we're going to let you get back to your Friday, and we'll see you in a few weeks. We have a session on community engagement, and research. Have a great weekend. Thank you, everyone.